Welcome to The Explainer. Today we are diving into one of the weirdest and, frankly, most dangerous things in all of biology, prions. Now, these aren't viruses, they're not bacteria, they're something else entirely, and we're about to break down exactly what they are and why they're such a unique threat. All right, let's just jump right in with a concept that sounds like it's straight out of science fiction. I want you to imagine a pathogen, a disease causer, with no DNA, no RNA, literally no genetic material at all. It's just a protein, a single rogue protein that can corrupt others in a devastating chain reaction inside the brain. And that's exactly what a prion is. The name itself, which was coined back in 1982 by Stanley Prusiner, is actually short for proteinaceous infectious particle. It's basically a corrupted, misfolded version of a protein that our bodies make all the time, especially in our nervous system. And this right here is the whole game. It's all about the shape. You've got the healthy protein on the left, PRPC. It's properly folded, it's soluble, it does its job, and when it's old, the cell breaks it down, no problem. But then you have the infectious prion on the right, PRPSC. It's misfolded, which makes it insoluble, so it starts forming these sticky, toxic clumps. And here's the kicker. It's basically a zombie protein. Any healthy protein it bumps into, it forces to misfold into its same deadly shape. So, once you've got this chain reaction going, how in the world do these rogue proteins get out of one cell and into the next to spread all that damage? Well, they pull off what is essentially a cellular heist. So it turns out, prions are smart. They actually hijack the cell's own internal mail system. You see, our cells are constantly releasing these tiny little bubbles called exosomes. Think of them like little cargo shipments used to send messages and materials to neighboring cells. Well, prions have figured out how to sneak aboard. And the process is just deviously clever. The prions get themselves packaged up inside these exosomes. The infected cell then sends them out, and these little Trojan horses travel to a healthy neighbor, fuse with it, and dump their prion cargo inside. And just like that, the infection has spread to a brand new cell. But, our cells aren't totally defenseless. They have this amazing internal cleanup system called autophagy. You can think of it as the cell's waste disposal and recycling crew. Its whole job is to hunt down, gobble up, and destroy junk. And that includes misfolded proteins like prions. And this is where it gets really interesting, because the cell can actually fight back. In experiments where scientists ramped up this autophagy system, say with a drug called rapamycin, the effect was incredible. The cell got way better at destroying the prions inside, which meant a lot fewer prions were able to escape in those little exosome getaway cars. On the flip side, what happens when that system fails? Well, when you shut autophagy down, the cell's defenses just crumble. The prions aren't being destroyed, so they build up and escape in massive numbers inside those exosomes. The infection just spreads like wildfire. And just when you thought it couldn't get any worse, it does. It turns out prions might have a secret accomplice. Hiding in our own DNA are these things called fossil viruses, basically ancient viral code left over from infections millions of years ago. And some research shows that if these get reactivated, they can actually supercharge the release of prions from the cell. Okay, so this isn't just a microscopic problem happening inside a body. Prions are terrifyingly tough. They can actually survive out in the environment for years, creating these hidden reservoirs of disease just waiting. I mean, just let this sink in. Infectious prions from the sheep disease scrapie were buried in soil, and they were still infectious more than 3 years later. This incredible durability is a huge part of why a disease like chronic wasting disease is so difficult to manage out in the wild. So, if these things are sticking around in the soil for years, it leads to a really unsettling question. Could a plant just growing in that contaminated soil actually pull the prions up into its tissues? And the answer, unbelievably, is yes. Studies showed that plants like barley and alfalfa can, in fact, suck up prions through their roots and transport them all the way up into the stems and leaves. And to prove it, when they fed those very plant tissues to mice, the mice got sick. This opens up a whole new and frankly terrifying potential pathway for transmission. Now, normally there's something called a species barrier. It's supposed to make it really hard for a prion from one species, say a sheep, to infect a totally different species like a deer or a person. But this barrier might be a lot more fragile than we'd like to think. Check out this wild experiment. Scientists took a screepy strain that they knew infected sheep with one specific genetic profile really well. Then they gave it to white-tailed deer. It infected the deer, no problem. But here's the crazy part. When they took the prion back out of the deer and put it into a new group of sheep, its behavior had totally changed. It had evolved. Now it was much better at infecting sheep with a completely different genetic profile. So what this means is that passing through an intermediate species acts like an evolutionary incubator. It can fundamentally change the prion itself. And the implications for a disease like chronic wasting disease, which is running rampant in deer, well, it suggests that prions could be changing and adapting as we speak. Which, of course, brings us to the biggest question of all. With chronic wasting disease spreading so fast and wide in deer, elk, and moose, could it jump to people? The scientific picture here is messy. For a long time, most studies in labs and on monkeys suggested the barrier was pretty solid. But then, a really important study in 2022 flipped things on its head. It showed for the first time that CWD taken directly from deer could, in fact, infect mice that were engineered with the human prion protein. But the way it infected them is what's really chilling. The mice didn't get the classic spongy brain damage doctors expect to see. Standard lab tests couldn't even detect the typical prion signature. Instead, there were these unique, smaller protein fragments. This is a huge deal because it means that if CWD did cross over to us, it could show up as an atypical disease that our current diagnostic tools would completely miss. And then there was this bombshell. The infected mice were shedding infectious prions in their feces. The researchers who discovered this put it plainly, saying, and I quote, "If this occurs in humans, it is a source for human-to-human transmission. That changes the public health risk completely." Up until now, we've talked about prions as an enemy from the outside, something you catch. But the final twist in this story is maybe the most profound. The enemy might also be coming from within. Some absolutely incredible research in 2021 found that our own cells, just through random mistakes made while reading our DNA, can accidentally create the very same mutant prion proteins that are known to cause inherited fatal prion diseases. It's not an infection, it's a typo that can create the seed of the disease from scratch. And that leaves us with a really provocative final thought. If our own bodies can spontaneously create an infectious protein just by making a mistake, and we know that even one single rogue protein is theoretically enough to start that fatal chain reaction, then what does a safe dose even mean? If the enemy can be generated from within, it challenges our entire notion of infection and risk.
Hello, and welcome to the Deep Dive, where we plunge into a stack of fascinating research to pull out the most important nuggets of knowledge. Today, we're tackling something both mysterious and, frankly, profoundly unsettling: prion diseases. These aren't just any brain disorders. They're caused by something so unique, it completely upends our understanding of infectious agents. Our mission for this deep dive is to explore the latest insights into what prions are, how they defy all conventional rules of biology by spreading in truly surprising ways, and the cutting-edge strategies researchers are developing to combat them. Get ready for some mind-bending facts and unexpected connections.
We'll definitely be navigating a complex landscape here. It spans cellular biology, environmental science, even genetics. Our aim is really to help you understand not just the core mechanisms of these protein-only agents, but also their broader implications for public health, and some truly surprising recent discoveries about how they operate. So as we prepare to dive into this complex world, where do we even begin to grasp something as fundamentally, well, bizarre as a prion?
Well, the place to start is often the beginning. What are these things that defy conventional biology? Because they truly are famously unusual. We're talking about prion diseases, also known as transmissible spongiform encephalopathies, or TSEs. They're a group of uniformly fatal neurodegenerative disorders affecting various mammalian species, including us humans. Think of specific examples like Creutzfeldt-Jakob disease, CJD, and Kuru in humans. Then there's scrapie in sheep and goats, bovine spongiform encephalopathy, that's mad cow disease, right, in cattle, and chronic wasting disease, or CWD in deer, elk, and moose.
And what makes prions truly unique, really sets them apart, is that the infectious agent, called PRPSC, it's just protein. It completely lacks genetic material like DNA or RNA, which is, what viruses and bacteria use to replicate. So instead of replicating like a living organism, Pathogenic prions multiply by a process we call structural information amplification.
Mm.
Imagine a perfectly good protein, the normal one, PRPC, suddenly encountering this misfolded rogue version, PRPSC. The PRPSC then acts like a kind of template, forcing the normal PRPC to also misfold into the pathological form. It's like a bad influence. It's sort of like a bad recipe that keeps copying itself perfectly without needing the original cookbook. And this PRPSC is also notoriously resistant to degradation by our body's usual cleanup enzymes, allowing it to accumulate pretty much unchecked.
Okay, so it's a rogue protein. It recruits normal ones to its side, and our bodies just can't clean it up. What are the visible consequences then? What happens when this protein rebellion takes hold?
Well, in the brain, the effects are devastating. You see what's called spongiform degeneration. The brain literally takes on a sponge-like appearance. Along with that, you get neuronal loss, the formation of these plaques made of the rogue PRPSC protein, and a type of brain inflammation called gliosis. For the individual, these lead to symptoms like ataxia, that's the loss of coordination, significant behavioral changes, and in humans, a really rapid and progressive loss of intellectual abilities. It's truly tragic. And this raises an important point about how these diseases actually arise. It's not just one way. They can be genetically inherited due to mutations in a specific gene called PRNP. That leads to conditions like familial CJD or Gerstmann-Straussler-Scheinker syndrome, GSS. They can also be acquired like variant CJD, VCJD, which was linked to consuming BSE-contaminated material, or Kuru, from those historical cannibalistic practices. But surprisingly, the most common form is sporadic CJD, which seems to appear spontaneously without a known genetic cause or external exposure. It's still a bit of a mystery.
And that risk of interspecies transmission, like mad cow disease jumping to humans, that's truly chilling, isn't it?
Absolutely. The consumption of BSE-contaminated material led to that VCJD outbreak in humans, tragically claiming 228 lives, mostly in Europe, especially the UK. But here's an even more concerning wrinkle. Even passage through an intermediate species can actually increase infectivity for humans. For example, research showed that BSE that had passed through sheep was found to be more efficient at converting human PRP in lab models than the original cattle BSE was. Which, brings up a key point. How many other intermediate species might allow for the genesis of new, maybe even more potent zoonotic strains capable of infecting humans completely under our radar?
That truly underscores the unique danger of prions. But the plot really thickens when we consider how these incredibly resilient prions defy containment. They persist not just within infected hosts, but out there in the environment itself.
That's right. Prions are unbelievably persistent and resistant to decontamination methods that would easily wipe out most other pathogens, like bacteria or viruses. This means they can remain infectious in the natural environment for extended periods, years potentially.
So they just stick around? How widely do they contaminate things? Are we talking soil, water?
Remarkably widely. Research has shown that prions efficiently bind to a huge variety of common environmental materials. We're talking soil, metal, plastic, wood, rocks, cement, aluminum, glass, even brass. And it doesn't take long at all. One study found that only 2 minutes of contact was enough for tiny plastic beads to become contaminated with enough prions to be detected and amplified in a lab assay. Just 2 minutes.
Wow. It's incredible how universally they bind. But okay, does binding to these surfaces actually mean they can transmit the disease from there?
It absolutely does. Infectivity remains, even after you try to wash and dry these contaminated surfaces. They're incredibly sticky. To test this properly, researchers created wire-shaped implants from various materials, polypropylene, aluminum, glass, stainless steel, even bits of rock. They contaminated these with prions, then implanted them directly into hamsters' brains. The result? A complete infection rate for most materials. Pretty much everything they tested led to disease. Interestingly, brass was the least efficient surface for transmission.
Huh. Brass. Why brass?
Well, that's a good question. We don't fully know yet, but it does raise the thought. What is it about brass that makes it different? Could that possibly offer a clue for future decontamination or prevention strategies?
Fascinating point.
Yep.
And to mimic more natural situations, did they just expose animals to contaminated surfaces in their environment?
Exactly. In another study, they did just that. Hamsters were simply exposed to large spheres, about 2 inches across, contaminated with prions, just left in their cages for 4 weeks. Many of these animals still got prion disease. They saw complete infection rates for wood, polypropylene, and cement spheres. The average incubation periods were around 6 to 7 months. Even stainless steel and aluminum spheres affected 60% of the exposed animals. So yeah, significant transmission. And transmission can occur through simple physical contact, like the animals rubbing against or licking these surfaces. What's even more intriguing, and maybe a bit alarming, is that the bedding material in contact with these prion-contaminated spheres also picked up the infectious agent and could then transmit the disease itself. So the environment itself becomes a kind of reservoir. Surfaces, bedding, it all becomes potentially infectious. But what about something even more, well, unexpected, like plants getting involved? That seems counterintuitive. It is truly astonishing, isn't it? But yes, plants can surprisingly act as vectors. Pathogenic prion protein, the bad stuff, can actually be taken up by plant roots from contaminated soil. And then it gets translocated throughout the plant, moving up into the aerial tissues like stems and leaves. This wasn't just in lab-model plants like Arabidopsis, it was also shown in agriculturally important crops like alfalfa and barley. These plants can accumulate enough prions from contaminated soil or media to transmit the disease, both through direct lab inoculation and, importantly, through oral feeding in mice, either forced feeding or just letting them eat the plant material.
That's a huge, completely unexpected pathway. Wow. Okay, if we connect this environmental persistence and these weird transmission routes, is there even such a thing as a safe dose of prions? Can you be exposed to a tiny amount and be okay?
That's the critical question, isn't it? And the current findings strongly imply that there is effectively no safe dose of prions. Research indicates a pretty clear linear relationship between even a low dose of prions and the probability of getting infected. This suggests there's no threshold dose below which the probability of infection drops to zero. Think about it like this. While higher doses generally lead to faster disease onset, for very low doses, the time it takes for the disease to show up might not be dramatically longer, but the chance of getting sick eventually is still there. It doesn't go away. This means even a minuscule, perhaps undetectable exposure could, in theory, eventually lead to disease down the line. It's a very sobering thought that completely redefines how we might need to think about exposure risks for these agents.
The idea of no safe dose is quite sobering, absolutely. Okay, but let's zoom in now from the wider environment to the microscopic world inside our own bodies. What's happening at the cellular level when prions invade? And what are researchers discovering about how we might actually fight back against this relentless protein?
All right, so at the cellular level, prion propagation, that misfolding process, is intimately linked to the cell's internal pathways for processing and degrading waste materials. The endocytic pathway, which cells use to bring substances in, and compartments like endosomes and especially lysosomes, which are like the cell's recycling center and incinerator, play crucial roles. They're involved in how PRPSC is trafficked around the cell, sometimes recycled, and ideally finally broken down.
So our cells already have these built-in recycling and cleanup systems. Does that mean we can somehow, I don't know, boost those systems to fight prions?
Exactly. That's a really promising area of research right now. The cell's own internal recycling and degradation system, called autophagy, is emerging as a key therapeutic target. You can think of autophagy as a cellular conveyor belt that moves waste, including misfolded proteins like PRPSC, to the lysosome for destruction. We talk about autophagy flux that just refers to the efficiency of this conveyor belt system, how smoothly and effectively it's running. And when this system is boosted, when autophagy is enhanced, it leads to more efficient delivery of PRPSC to the lysosome for degradation. It helps clear the bad stuff out. This provides a really clear therapeutic target. And indeed, pharmacologically inducing autophagy, using drugs to ramp it up, has already provided solid experimental proof of concept. We've seen it work against prions in both cell cultures and mouse models. What's more, increasing autophagy also seems to reduce the release of prions and exosomes, those little communication vesicles, which helps limit how they spread between cells.
That's incredibly promising. So autophagy is a key target. What specific compounds are researchers actually looking at? Have any shown particular promise in the lab, even preliminary?
Yes, several compounds have definitely shown promise in manipulating autophagy to fight prions. It's an active area. For example, there's imatinib. It's actually a cancer drug, but it was found to prolong the survival of prion-infected mice by a significant average of 17 days. The caveat there was that its effects were less pronounced once the infection was really established in the brain, probably because imatinib doesn't cross the blood-brain barrier very well.
The blood-brain barrier, always a hurdle.
Always a challenge in neurodegenerative disease, yeah. Then there's rapamycin. This one is a well-known autophagy inducer. It also extended survival in prion-infected mice by an average of 13 days in one model. And in a mouse model of GSS, that inherited form, it delayed disease onset and prolonged survival by an average of 16 days. So these studies suggest rapamycin might have a better chance of actually getting into the brain and doing its work there.
Okay, imatinib, rapamycin. What about other compounds? Were any particularly effective maybe in cell cultures first?
Definitely. Another one called AR12 or OSU0303RELV showed really striking results in cell culture. It completely eradicated prion infection in persistently infected cells after just 2 weeks of treatment, which is pretty remarkable.
Wow, eradicated. How does it work?
It seems to work by kickstarting a specific cellular stress pathway that induces autophagy, essentially supercharging the cell's internal recycling system to clear out the PRPSC. When they tested it in prion-infected mice, giving it intraperitoneally, it extended their survival by an average of 11 days, so promising translation from cells to animals too. There's also spermine, which is actually a natural compound found in our bodies. It's found to clear PRPSC in cultured cells, again by inducing autophagy, possibly by letting another protein called TUB6 interact with the prion aggregates for removal. And FK506, also known as tacrolimus, an immunosuppressant drug. It decreased PRPSC levels in multiple cell models and prolonged survival by 16 days in prion-infected mice. Lastly, trehalose, a type of sugar, showed some ability to counteract prion infection in cells and decrease the prion load in the spleen of mice, but it didn't significantly prolong survival when given orally. So you see a range of compounds, different mechanisms maybe, but many point towards autophagy as a really powerful lever we might be able to pull therapeutically.
That's a fascinating list. It really underscores the potential of targeting autophagy. But this raises an important question you touched on earlier. How exactly do prions spread between cells in the first place? And how does autophagy fit into that picture of cell-to-cell spread?
Right, that's crucial. It turns out PRPSC is closely associated with exosomes. These are tiny vesicles, like little bubbles, that cells release to communicate with each other or get rid of waste. Studies have detected significant prion conversion activity, meaning the ability to spread them as folding inside exosomes isolated from prion-infected cells. The infectivity found in these exosomes was comparable to what you find in brain tissue from terminally ill animals, so they're potent carriers. And if you stimulate cells to release more exosomes, for example, by treating them with a compound like monensin, it actually increases the intercellular transfer of prions between cells. Monensin also seems to hinder the normal processing of the good prion protein, PRPC, leading to more of the misfolded PRPSC version over time. So it's a double whammy. But crucially, and this connects back to our previous point, enhancing autophagy actually decreases the release of prions in these exosomes. They observed that cells with naturally less efficient autophagy machinery release significantly more PRPSC and prion infectivity via exosomes compared to cells with more robust autophagy. This indicates a really intricate crosstalk between the autophagy cleanup system and the exosome release pathway. It directly influences how prions spread laterally between cells. And these exosomes can even mediate prion transfer between cells from different species, highlighting another way interspecies barriers might be crossed.
That's incredible. So the cell's own machinery for cleanup, autophagy, and for communication, exosomes, are playing these absolutely vital roles in both spreading and potentially stopping prions. But what about even smaller, maybe more unexpected players? I feel like I've heard some buzz recently about things like microRNAs and even ancient viruses being linked somehow to prion-like spreading. Is there anything to that?
You're absolutely right to pick up on that. It's a rapidly evolving area. Let's start with microRNAs, or miRNAs. These are tiny RNA molecules, just little snippets of genetic code, maybe 1825 nucleotides long. Cells produce them to fine-tune gene expression after the main genetic message has been made. Think of them like cellular dimmer switches, regulating how much protein gets made from a specific gene's RNA message. Now, this is significant for prions because some miRNAs can directly target and turn down the expression of the normal cellular prion protein, PRPC, by binding to its messenger RNA. And decreasing the amount of normal PRPC available is key, right? Because that hinders the amplification of the bad PRPSC form. Less fuel for the fire. For instance, one large-scale screen identified 13 different miRNAs that were capable of directly regulating the production of the prion protein.
So these tiny RNA regulators are definitely involved in the prion story. That sounds like another potential therapeutic avenue right there, doesn't it? Targeting PRPC levels.
Exactly. And going further, when researchers looked at prion-affected brains, they found distinct patterns of miRNA deregulation. Some specific miRNAs were consistently upregulated, while others were downregulated compared to healthy brains. This naturally leads to the question, could we develop therapies based on either replacing the missing miRNAs or inhibiting the overactive ones? There are various strategies being explored, like using chemically synthesized mimic miRNAs for replacement, or things like anti-miRNAs or miRNA sponges to soak up the problematic ones for inhibition. However, it's definitely not straightforward. There are significant challenges. One major concern is off-target effects. where an artificial miRNA might accidentally interfere with other important genes. Plus, giving high doses could potentially saturate the cell's natural miRNA processing machinery, disrupting normal biological functions. And another complication is that some miRNAs show fluctuating expression levels as the disease progresses, which makes therapeutically fine-tuning their levels really quite challenging. It's complex.
Okay, so microRNAs are involved. Complex, but potentially targetable. But what about viruses? That seems like a really surprising connection for what we think of as a protein-only disease.
This is truly fascinating and a relatively recent area of discovery. Evidence is accumulating that endogenous retroviruses, or ERVs, play a role. Now think of ERVs as ancient viral fossils buried deep within our own DNA. They're remnants of retroviral infections that happened millions of years ago and became integrated into our germline DNA, passed down through generations. Normally, these ERVs are epigenetically silenced, basically kept switched off by the cell. But studies are finding that they can become reactivated and upregulated in various neurodegenerative diseases, including potentially prion diseases.
Wow, ancient viral remnants waking up inside us. That sounds like science fiction. And how does that relate to the spread of misfolded proteins like prions?
It's pretty wild, right? Studies, initially in mouse models, uncovered the activation of these endogenous retroviruses dramatically increases the intercellular spreading of what they call proteopathic seeds. These seeds can be composed of various misfolded proteins, like a yeast prion domain used in experiments, or even pathological human tau protein aggregates, which were involved in Alzheimer's disease. The viral envelope protein, the N protein from the reactivated virus, seems to play a particularly prominent role in helping these protein aggregates spread between cells. This process often involves those extracellular vesicles, the exosomes we talked about earlier. They get loaded up with the protein aggregates and decorated on their surface with this viral env protein. The efficiency of this aggregate spreading then depends on the specific interaction between the viral env protein on the vesicle and its corresponding receptor protein on the recipient cell surface. What's truly compelling, and brings it closer to home, is that subsequent studies found that expressing human endogenous retrovirus ENV glycoproteins from various ADRV families like Agiu-Vabiu, ERV-K, and others also significantly increased the dissemination of protein aggregates between human cell cultures and even in primary human brain cells called astrocytes.
So our own ancient viral DNA might be actively helping these toxic proteins spread in our brains.
That's what the evidence is strongly suggesting. It points towards a completely novel therapeutic direction too, using antiviral drugs. Specifically, viral protease inhibitors, like a drug called Emprenavir, have been shown in these studies to actually reduce this ERV-mediated spreading by blocking the maturation of the viral proteins needed for the process. It's a huge potential aha moment, really.
Yeah.
It completely reframes our understanding of how misfolded proteins might spread, suggesting that these normally dormant ancient viral elements could be acting as accelerators for a whole range of neurodegenerative conditions, maybe including prion diseases.
That is a truly profound revelation. Our own ancient viral remnants potentially accelerating the spread of these harmful proteins. Just incredible. Oh, but let's take it one step further. What if the very source of some of these misfolded proteins isn't always a clear genetic mutation passed down through families, or an external infection like eating contaminated meat? What if it's just a simple mistake, an error happening during a really fundamental biological process within our own cells, day in and day out?
This leads us perfectly to one of the most intriguing and perhaps unsettling recent insights in the field. The idea that, quote, transcript errors generate a continuous stream of amyloid and prion-like proteins in human cells. We know that aging is characterized by the accumulation of these misfolded, often amyloid or prion-like proteins in various tissues, especially the brain. New research is demonstrating that errors occurring during transcription, that's the crucial process of copying the genetic information from DNA into messenger RNA, can actually generate these problematic amyloid and prion-like proteins. And this isn't just a theoretical possibility. They've observed it happening in various human cell types, including embryonic stem cells, developing brain organoids, and fully differentiated mature neurons.
Hold on. So our cells are basically making typos when they copy the DNA instructions into RNA, and some of these typos can directly result in the creation of these disease-causing misfolded proteins.
Precisely. It's like a subtle, ongoing source of potentially toxic proteins being generated internally. And what's even more astonishing, perhaps, is that some of these erroneous mutant proteins generated by these transcription errors are identical to those previously identified in familial cases of amyloid diseases, cases we always assumed were caused solely by inherited mutations in the DNA itself. For instance, researchers found a specific transcript error that generated PRMPA133V, that's a mutant human prion protein known to cause the inherited prion disease, Gerstmann-Streussler-Scheinker syndrome, or GSS. Other transcription errors they detected generated mutant versions of proteins that closely resemble those implicated in familial forms of CJD. This really challenges a long-held assumption, doesn't it? It raises a fundamental question. Could both familial cases, caused by inherited DNA mutations, and non-familial or sporadic cases of these amyloid diseases actually be caused by the very same mutant proteins, the only difference being the mechanism by which that protein is generated, a permanent fixed genetic mutation in the DNA versus a transient random error during RNA transcription?
That is a potential paradigm shift. It implies that even without inheriting a specific genetic defect, our own normal, albeit imperfect, cellular processes could be accidentally creating the very proteins that lead to these devastating neurodegenerative diseases just through random errors.
Exactly. It suggests an intrinsic vulnerability, and the plot thickens even further when you consider what might increase these transcription errors. DNA damage, which is a well-known hallmark of human aging and is also a feature of multiple proteotoxic diseases like Alzheimer's, has been shown to readily generate these transcription errors. More DNA damage leads to more mistakes in the RNA copies, and perhaps ironically, even some cutting-edge therapeutic approaches being explored, like specific types of RNA editing designed to fix genetic diseases, could potentially inadvertently create these kinds of erroneous transcripts if they're not perfectly targeted. It really suggests a fundamental internal source of potentially pathogenic proteins that we've only just begun to fully appreciate and understand.
Wow. We've truly taken a deep dive today, uncovering just layers upon layers of complexity and so many surprising connections in the world of prion diseases, from their unique protein-only nature and how they defy biology, to their astonishing persistence out there in the environment, and then zooming right into the intricate cellular battles being waged against them.
And we've seen how modern research is uncovering these completely unexpected players in the story, from the subtle but potentially powerful regulatory role of microRNAs to the genuinely surprising role of reactivated ancient viruses, possibly accelerating protein spread between cells. And even the fundamental process of transcription itself, the simple act of copying our genes, potentially contributing to the very proteins that cause these devastating diseases through unavoidable errors. It really makes you think about all the hidden forces at play deep within our biology and just how complex and interconnected everything truly is.
It really does. This knowledge challenges us to think very differently about neurodegeneration. So for you, our listener, here's a final thought to chew on, building on everything we've discussed. Given that even very low doses of prions appear to have no truly safe threshold, and that our own cells seem to be constantly, inevitably generating potentially misfolding proteins through transcription errors as we age, what fundamental shifts might be necessary in our approach to understanding, preventing, and maybe one day treating neurodegenerative diseases? Shifts that go beyond just targeting inherited mutations or looking for external infectious sources. What does this internal persistent vulnerability mean for the future of brain health?